Hi everybody, and thank you for listening to my presentation today. So I'll be talking about my research which focused on the effects of multiple stressors on the ecophysiology of Lampsilla silicoidea, namely looking at the effects and interactions occurring among water temperature, velocity, and suspended solid concentration. So to start off, unionids or freshwater mussels provide beneficial ecosystem services to freshwater systems by way of creating habitat, recycling nutrients, coupling the energy between the benthic and pelagic zones, and depositing organic materials. However, recent threats to freshwater systems have presented themselves as slow, chronic stressors that can affect mussel ecophysiology. A stressor is known as a condition, event, or variable that exceeds its normal range of variation and can have a negative effect on an organism. And unfortunately, nearly 70% of North American unionid species are listed under the Species at Risk Act. So in order to understand the effects of the multiple stressors, we must first understand what's going on individually. So in terms of temperature, temperature has been linked with increases in both CR and OC, CR being clearance rate and OC being oxygen consumption rate. It's also important to mention that when I say temperature here, I'm talking about acclimation temperature response, which is an animal's response to a gradual change in temperature. And the alternative would be talking about the acute temperature response, which I'll touch on later on in this presentation. So next, with TSS concentration, previous studies have found that TSS is linked with negative effects on both CR and OC. And finally, velocity has been found to increase both CR and OC. So now that we understand the individual effects, we can go on to look at the interactions occurring when we put two of them together at the same time. Some interactions we may be able to see are things like synergism, when both stressors are working to push the response variable in the same direction, or antagonism, when the stressors are working against each other and pushing them in opposite directions. We may also see additive or multiplicative effects, which is when we compare the sum of the pooled stressor effects on the response variable to that of just the sum of one stressor on its own, and we compare those, and if we find that the sum of the pooled effects is greater, we call this an additive effect. And likewise, if we took multiplication instead of addition, we would call this a multiplicative effect. So now in the terms of what's found in this Venn diagram, if we go back to temperature and TSS concentration, these two stressors together have antagonistic effects on OC, while we're not quite sure what happens with CR just yet. So this indicates that the negative effects of TSS concentration are ameliorated by the positive effects of temperature. Likewise, we see that TSS concentration and velocity also have antagonistic effects on CR, while we're not quite sure what happens with OC. And yet another gap that remains to be addressed is what happens when we put temperature and velocity together. And where my research came in was I wanted to know what happened when we looked at all three stressors on their own. So in order to go about this, I went out into the field and I collected 51 male fat mucket mussels, and I housed them in the Hagen Aqualab in flow-through chambers that you can see pictured here on the bottom right image, and they were held until the end of fall in 2019. And I conducted 1-hour trials where I measured oxygen concentration, temperature, velocity, turbidity, and fluorescence in these flow chambers. I was able to maintain temperature through either heating or chilling the water as needed in an outer water jacket, and the mussels were acclimated in sediment chambers for about 20 minutes before trials started. And finally, all of this was run simultaneously alongside a no-mussel control trial with paired shells, and I was able to calculate CR, OC, and scope for growth rates. Now my main experiment, called my multi-stressor experiment, was devised into a split-split plot design, where although it looks a little complicated here, it really just comes down to there being 3 factors with 3 levels each. So factor A was acclimation temperature, whose levels were 12.5, 20, and 27.5 degrees Celsius. Next, factor B was velocity, where the levels were 0, 15, and 25 centimeters per second. And lastly, factor C with TSS concentration had levels of 0, 10, and 20 milligrams per liter. This created 27 possible combinations where I did 5 replicates for each, and I was able to measure clearance rate and oxygen consumption rate for each one. For my second set of experiments, I wanted to compare the effects of acclimation and acute temperature. So here, I acclimated mussels in my acclimation temperature trials to either 12.5 or 27.5 degrees Celsius, and then tested them at that same respective temperature. On the hand of acute temperature trials, I would acclimate mussels to those two temperatures, but then test them at a new temperature of 20 degrees Celsius. And using those results, I was able to calculate the rate of change of the acute response, which is the rate of change in oxygen consumption as a consequence of changing the temperature. And all of this was done at 3 levels of TSS flux, which were 0, 150, and 500 milligrams per meter squared per second. So let's start with our clearance rates in our multi-stressor experiment. So to start off, I found that CR followed a unimodal response to temperature, where although it wasn't statistically significant, CR appeared to increase from 12.5 to 20 degrees Celsius and then decrease from 20 to 27.5 degrees Celsius. Next, with chamber velocity, CR increased significantly from 0 to 15 centimeters per second and then decreased significantly from 15 to 25 centimeters per second. And last of all, with TSS concentration, CR decreased from 0 to 10 milligrams per liter and plateaued thereafter. So now let's take this one step further and break it down. So with chamber velocity along the x-axis, we can see here on our bottom graph that we are looking at the individual effects of CRs occurring at 20 degrees Celsius. So from 0 to 15 centimeters per second, CR is increasing in every given case of TSS, and from 15 to 25 centimeters per second, CRs are decreasing in every given case of TSS. And what we found here when we compare it to the pooled effects found above, which is taking all of the effects together, we found multiplicative effects to be present from 0 to 15 centimeters per second at 10 and at 20 milligrams per liter TSS. So this indicated that the change we see from 0 to 15 centimeters per second in the pooled effects, that is all the temperatures and TSS levels put together, this change was greater than the change occurring at 10 and the change occurring at 20 milligrams per liter. That's the orange and the green lines. However, this wasn't the case in the case of the blue line, which is 0 milligrams per liter TSS, nor did it occur from 15 to 25 centimeters per second in any given condition. So next, let's head over to 12.5 degrees, where here, once again, along the x-axis, CR increased from 0 to 15 centimeters per second, although to a relatively lower extent than what it did at 20 degrees Celsius. And then from 15 to 25 centimeters per second began a slight decline, except for the case of 0 TSS, where it continued a slight incline. And here I found multiplicative effects to be present with all velocity changes and at all levels of TSS. So again, what this meant was that from 0 to 15 centimeters per second, the changes we see here were greater when they were multiplied than any of the changes individually seen here when they were multiplied. And likewise, the same thing from 50 to 25 centimeters per second was found, where these changes were greater than any one of these single changes. Last of all, let's head over to 27.5 degrees Celsius, where I found that with chamber velocity, CRs increased from 0 to 15 centimeters per second, with a relatively steeper increase in the case of 10 milligrams per liter TSS, which was an interesting result, as well as a more dramatic decrease when velocity increased up to 25 centimeters per second. And here I found additive effects to be present this time. This occurred at higher levels of TSS with both velocity changes. So last time we'll go through it, but essentially from 0 to 15 centimeters per second in our pooled effects, this change ended up being higher than any of this change, the orange line at 10 milligrams per liter, and this change, the green line at 20 milligrams per liter. And this also was the case from 15 to 25 centimeters per second, where this change was greater than the individual change at 10 and the individual change at 20 milligrams per liter, not happening, however, at 0 milligrams per liter TSS. So that just about concludes clearance rates. Now let's hop over to oxygen consumption rates and scope for growth. So OC, oxygen consumption, increased significantly from 12.5 to 20 degrees Celsius and plateaued thereafter. Also, OC increased significantly from 15 to 25 centimeters per second chamber velocity. And with TSS concentration, OC appeared to decrease from 0 to 10 milligrams per liter and increase from 10 to 20 milligrams per liter, although these results were not significant. And in terms of scope for growth, or SFG, SFG followed a unimodal response to temperature, where it appeared to increase from 12.5 to 20 degrees Celsius and decrease from 20 to 27.5 degrees Celsius. Next, it decreased significantly from 15 to 25 centimeters per second. And finally, while it wasn't statistically significant, it appeared to decrease from 0 to 10 milligrams per liter TSS and plateau thereafter. In the case of the oxygen consumption model, no significant interactions were found in the split plot ANOVA, as well as in the scope for growth model. So I didn't go into any further analyses regarding any presence or absence of multiplicative or additive effects. Now for my secondary set of experiments where I compared acclimation versus acute temperature effects, I was able to calculate the acute rate of change, and here I found that mussels that were acclimated to 12.5 degrees but then tested at 20 degrees, so they were suddenly warmed, had an acute rate of change of nearly 4, which would indicate that their oxygen consumption is increasing by a factor of 4. And on the other hand, the mussels that were acclimated to 27.5 degrees but then tested at 20 degrees, so they were suddenly cooled, had a rate of change of about 1, which would indicate that they're changing by a factor of 1. So they're not changing at all, really. So that was definitely an interesting result to find. And next, I found with clearance rates that mussels that were acclimated to their test temperature had significantly higher clearance rates than those that were tested acutely. And also, the mussels that were acclimated to 27.5 degrees had significantly higher clearance rates than those that were acclimated at 12.5 degrees. And finally, for scope for growth, I found that mussels that were acclimated to their test temperature had significantly higher scope for growth rates than those that were tested acutely, specifically when speaking about those that were acclimated to 12.5 degrees and then tested acutely. So with the results of this study, we were able to confirm previous findings of individual stressor effects, as well as look into more detail as what's happening when we put two stressors together and look at those combined effects. And that's important because in nature, environmental factors aren't acting individually, they're all acting at the same time. And lastly, we were also able to fill in the blank of what's happening when we look at all three stressors at the same time. And the answer came down to, it depends on acclimation temperature. Going further into that, I did find that the primary stressor affecting CR changed depending on acclimation temperature. For example, at 12.5 degrees, I found that velocity and TSS acted synergistically to reduce CR, and the multiplicative effects here indicated that this reduction was larger at this temperature than at the others. So here, temperature acted as the primary stressor. At 20 degrees, the decline in CR from 15 to 25 centimeters per second was larger at this temperature than at the others, because the single effect of velocity was greater than the pooled effects. So here, velocity acted as the primary stressor. And finally, at 27.5 degrees, the reductions in CR we saw at 10 and 20 milligrams per liter TSS were larger at this temperature than at the others, and these were additive. So here, TSS concentration acted as the primary stressor. Now, while it's important to remember the intensification of these three stressors, it's also important to remember that the acute changes in temperature also have significant effects on mussel feeding, respiration, and scope for growth. Forecasted increases in precipitation, temperature, and sediment loads will have serious deleterious effects on mussel feeding, and I can forecast a worst-case scenario being something like a high TSS event occurring in late summer when temperatures are at their highest and flows are at their lowest. And an example of this would be something like a riparian disturbance occurring at that time. It may sound dire, but it is important that we recognize the effects of multiple stressors in order to help inform regulations and SAR recovery plans. And ultimately, I found the multi-stressor approach to be a more ecologically relevant approach, and I recommend it for study with other additional environmental conditions. And on that note, I'd just like to thank everybody mentioned on this slide. I'd like to thank all the organizers for this FMCS symposium and all of you for listening to me here today. Thank you.